Thanks to all of you for coming and to the local organizers for helping put on this conference, and Society for Social Neuroscience for this conference. So as Jean mentioned, I'm at Caltech, which is very sort of basic research oriented, very heavily in physics, engineering, biology as well, but it doesn't actually have a psychology department at all. So I'm kind of a bit of an unusual lab there, trying to bridge basic biological approaches with psychological questions. And so the topic today, this is our lab is interested in social neuroscience very broadly. And we look at many different brain regions. We use many different techniques, and I'll tell you about some of those in the next hour. And we study a number of clinical populations, and one of those is autism. And you've already heard a little bit about it, and you'll hear more about it in the course of this conference. So the title of the talk here is going to be Social Brain and Autism, and it'll focus on autism, but make some comparisons also to other patient populations that we've studied, and give you a sense for the different approaches that my lab has used. So to give you a preview, actually this could be the concluding slide, of what ties together several themes of research in my lab with respect to autism. So the first one is that, as we now know, social cognition depends to some extent on a network of specific brain regions, the so-called social brain. So it's a collection of brain regions that do lots of things and that may not be only processing social information, but nonetheless there's evidence that many of them may be to some extent specialized for processing social information. And one of these brain structures that my lab has studied in a lot of detail is the amygdala. And I'll show you a little bit of that data as I go on in my talk. So, as I mentioned, there's these structures, the amygdala, the insula, the ventromedial prefrontal cortex. You've heard about several of these already in some of the talks this morning. But they communicate with one another as components of a network. So the amygdala doesn't do anything in isolation. It's part of the extended network. It's input from some regions of the brain, for instance, ventral temporal neocortex that gives it information about faces, has connection with reward structures, and so forth. And so to investigate these, there are a lot of techniques available. In humans, they're fairly indirect and depend on MRI techniques. But so resting state and functional connectivity, MRI techniques, which is what those words up there mean, are often used to look at the strength of interaction, the connectivity between these brain structures. So we have a set of brain structures, we have tools available for looking at their interaction, and of course they interact in part because they're actually connected to one another with white matter tracts, with axons. That's much harder to look at in the human brain, although there are techniques for it. But one population, one clinical population that I will be telling you about a little later in the talk, actually at the end of the talk, are people who are born without the biggest white matter tract in the human brain, that is the corpus callosum. So people born without the corpus callosum are one model to look at what happens, what the role of these white matter connections is in social networks. And then what ties all of this together is that one leading hypothesis of autism at the systems level, as you may know, is that it involves dysfunction of long-range connectivity in the brain. So it's abnormal strengths of connections between many of the structures that mediate social cognition in the brains of people with autism. So just to summarize, this is sort of a preview of some of the broad background and the way in which these are thought to motivate research on autism that I'll be telling you about in the next hour. And so here's the menu of what I will be showing you. I will begin with just some behavioral. First there'll be a little introduction and review of some things. Then I will tell you about eye tracking studies in people with autism. Then about direct recordings through electrodes in surgical patients from the brain in people with autism, an fMRI briefly, and then make some comparisons to other clinical populations. So to begin with, here's a somewhat simplified and to some extent outdated view of the social brain. So these colored regions, the names of which are all listed here on the left, are many of these components of the social brain. So these are structures that are important for processing social information. If you do fMRI studies that look at social cognition, components of social cognition, you activate many of these brain structures. If you record, mostly in animal studies or in rare human cases from these regions, you find that cells respond to social stimuli like faces. If you lesion these structures, like the amygdala, which is the one right back here, you can cause fairly selective impairments in aspects of social cognition. So there's lots of evidence implicating these structures. And again, a big caveat being that these are all connected together, so they don't function in isolation, but they're components of a brain network. But just so this orange region here, and this coronal section of the human brain, so if you cut the brain, like along this line here, then you would see the amygdala in the medial temporal lobe here. And that's one structure, as I mentioned, that my lab has been studying in a lot of detail. Quick background on autism, with which many of you are probably familiar. So as you know, this is a psychiatric illness that is a pervasive developmental disorder, which means pervasive means that you have it typically lifelong, so you don't grow out of it. Developmental disorder means that it develops fairly early in life. So you aren't typical, and then suddenly at age 20 you develop autism, but it develops fairly early in life. It's diagnosed around age 3 years, but there are some markers already prior to a full psychiatric diagnosis. So it's a pervasive developmental disorder with an early onset, and you have it lifelong. There's a very large genetic component. There are many family studies done, going back to original ones by Michael Rudder and colleagues, that show that it's actually very highly heritable. So autism has a big genetic component, not 100%. There are also environmental factors, but a big genetic component. And one additional evidence for that is that there's a big ratio in favor of males, about 4-to-1 in general, 4 times more likely in males than in females. What's important to point out is that even though lots of people study the genetic basis of autism, there's no single gene for autism, or even a small number of genes for autism, but rather many hundreds of genes that all contribute to the risk of developing autism. A few of these genes, like those involved in some other disorders, like Fragile X syndrome or tuberous sclerosis, increase the risk a lot, but most of them contribute a very small portion of the risk. And what people are looking at now is, what is it that ties together all of these different genes? And the answer seems to be is that they participate in a few gene networks, for instance, responsible for synapse maintenance and strength. And if that goes awry, you have functional problems in communicating between different brain parts. And one other important point is that autism is a spectrum, so it's recognized, has been recognized for a long time, and is now in DSM-5 explicitly recognized, so that there's a broad range that is continuous with the distribution of behaviors relation. So it's a psychiatrically defined illness, and even though there are many biological markers, currently it's based just on behavioral criteria. So just to orient you with respect to very broadly what's known about autism. So as I mentioned, there's a big influence of genes, many of them, hundreds, that interact, and their protein products influence lots of different things in the brain that interact with environmental influences and eventually give rise to problems in the synapses and the way in which brain regions can communicate. And then what we finally measure is the altered behavior and cognition. So where my lab has been working has been sort of at this level, looking at the behavior and cognition in people with autism, and then asking some systems level questions about the brain. Presumably all of this based on these lower level descriptions, which my lab doesn't investigate, but most other labs working on autism do. The criteria, as you may know, are this triad. So autism was diagnosed, and to a large extent basically still is, in these three basic domains. So social impairments, interacting, reciprocal social interactions with other people, communication deficits like delayed onset of language, and then also stereotyped interests and repetitive kinds of behaviors. These, to some extent, the population that my lab studies, they are all high-functioning adults with autism for a number of reasons. One is just practical, that they can do all the cognitively demanding studies that we have, and that population has a disproportionate impairment in this first domain. So they're really impaired in social interaction. So the reason that my lab studies autism is not so much because I have a family member with autism or I really want to cure autism, although I hope that the research contributes to that, but it's more as a model for understanding social cognition and doing basic social neuroscience research. And so you have this disproportionate impairment in social functioning, especially in high-functioning individuals with autism. And just very briefly, these criteria are, to some extent, now outdated officially, because these criteria, the psychiatric criteria that were in the Diagnostic and Statistical Manual of the American Psychiatric Association, edition 4, so this is how it's diagnosed, has been superseded by the 5th edition of this, which is actually very similar, and it contains this very similar criteria, even though it doesn't officially make some of the same distinctions, like between Asperger's syndrome and autism, as the previous one did. But more importantly, there's now a big push amongst research scientists, not psychiatrists diagnosing autism, but those of us doing research on autism, to recognize that really this isn't an entity that should be defined as a syndrome behaviorally, but you want to give a more dimensional description. And so that's one thing that many people are doing. I just have this quote here from Tom Insel to make that point, where he says it's critical to realize that we can't succeed if we use these psychiatric categories, the original DSM categories, but rather we have to base our diagnosis of psychiatric illnesses like autism on the emerging research data rather than the current symptom-based categories. And so in that spirit, that's a lot of the research that we are doing. So let me go into the research that we're doing. One big question that we're looking at is what is it that motivates us to pay attention to social stimuli, that makes social stimuli rewarding. And linking this to autism is the hypothesis that if you disrupt these kinds of social attentional or motivational processes early in life, so if you have a baby that is going to develop autism, but the baby isn't interested in looking at faces or interacting normally with its mother or later with other children, then what you would have as a consequence of that during development is reduced social input to the brain. If the baby isn't interested in looking at faces, the brain isn't getting information, getting input about faces, and as a consequence of that, you would have abnormal, reduced development for specializations, for face processing, for theory of mind mechanisms where we infer people's emotions and intentions from looking at them, and so forth. And of course, this would be kind of a vicious cycle. So the idea is that if you had even a rather small deficit early in development in social motivation, this could sort of amplify as the baby begins to grow older and older, could amplify into a developmental path that ends up being autism. So there are many ways in which you could imagine looking at this mechanism. The one that we focused on is faces. And there's many good reasons for that, but right now, if you ask yourself, what are you looking at mostly? You're looking, and we did eye tracking on you, what are you paying attention to? There are two things you're looking at mostly. One is my face, highly salient, and the other is text, which is very derivative. But text and faces are the most salient stimuli. And if you have those, people will pay attention and look at those. Not just people, but here's a video of a baby monkey so they're much more precocious than humans at birth. But this baby monkey here, what's striking about the behavior of the baby monkey is that it is very interested in looking at the face of the experimenter. This is a bit of an artificial experiment. There's no distracting stimuli and so forth. Nonetheless, it makes the point that all primates are very interested in looking at conspecifics and looking at faces. And the reason is simple. The reason you look at faces of other people is because they carry important social information. If somebody has a certain facial expression, if somebody is looking at you or looking somewhere else, those are important social cues that you need to pick up. We can measure this and quantify it very accurately in humans using eye tracking. And so nowadays we do this without having to put anything on the head. But this is just to give you the picture of how it would work. You would have little video cameras here that basically measure the direction of gaze, where the person is looking. As I mentioned, nowadays you don't need to wear a head-mounted eye tracker, but the principle is the same. You look at often the corneal reflection of infrared beam, and you figure out where is the person looking when they're looking at a face, for instance. And when you look at a face, a photograph of a face, you look at it more or less like is shown here. So you look around like this little spotlight. You make 3 or 4 or 5 fixations per second, and you explore the face with your eyes, and you preferentially look at those regions of the face that carry the most information. And those are the regions that can change the most, which is the eyes and the mouth. So most people look a lot, in particular at the eye region of the face. So let me jump to the first data slide. It has a couple of different data points on it, so let me just walk you through this here. What is shown here, these are maps that show how frequently people look at a certain part of a face. So the more hot the color, the red color here means that people look most at this eye region of the face. And we have these eye tracking data here for 4 groups of people: matched controls, people with autism, and you see that the controls look at the eyes a lot. People with autism look less at the eyes, and you can see that most easily here in just the difference. If we just subtract these two, the red is where the controls look most, and the blue is where the autism subjects look the most. So this finding has been replicated also by many other groups, the finding that people with autism do not look at faces normally. They do not show the normal preference for looking at the eyes. And one interesting comparison is that a subject that we've been studying for a long time, and I won't show you any more data from her, but this is a subject who has damage to the amygdala, one of these key regions of the social brain, and she's somewhat similar to what you see in autism. Like people with autism, she also doesn't look at the eyes in faces normally. So for some time now, and this is work by 2 people down here who were postdocs in my lab when they did the work. So for some time we've had 2 pieces of information. One is people with autism do not look normally at the eyes and faces. So there's something abnormal about face processing in autism. And the second piece is that this abnormality in autism looks similar in some ways to what you find in a patient who doesn't have autism but has just focal lesions of the amygdala. So that implicates the amygdala as maybe one structure that contributes to the abnormal social behavior in autism. So we would like to look at that in more detail. And I'll show you some of those data a little later. I just wanted to show you, because I will show you more data from the same task shortly, a slightly different way of looking at this information. So what is shown here from a paper by Frederick Gosselin, Philippe Schins, and colleagues, is this is not now a map of where people look at faces, but this shows the information that people make use of from a face, in this case to tell apart the different emotions in a face. So these results, taken from this paper down here, show that people make a lot, you would make a lot as viewers, would make a lot of use of the mouth region of the face, the smile, to figure out if somebody is happy, of this to figure out surprise, and you make a lot of use of the eyes in particular for the emotion fear. How do you get these kinds of pictures? Let me just briefly describe to you the methods for this. It's called bubbles, developed by Fred Gosling and Philip Shins. And what you do is you show people faces, but only small parts of a face. So let me show you just how that experiment works. If you were a participant in this experiment, I would show you one of these faces here and ask you to push a button to tell me, is this happy or is it fear? Okay, so if you did that for these faces, happy or fear, each of you would be able to tell this is fear, this is happy, and you would be at 100%. But we make it difficult for you because we don't show you the whole face, but we only show you little pieces of the face. So on each trial, you would be sitting there, you have to push a button now and tell me, is this happy, is this fear? But what you see looks like this. A little bit of an eye or a mouth. On the next trial, you would see this. On the next trial, you would see that, and so forth. So on each trial, randomly, you would see very small parts of these 4 faces that I showed you. Now you're no longer at 100%, but you're more like 70% or so. So you get some errors. And what we can do, if we show you many of these stimuli, is we can put together, we can add up all those trials, all those little pieces of faces, where you were correct and you were able to tell this was happy and this was fear. And we subtract from that all those trials, all those pieces of faces where you were incorrect. And if you do that, that's how you get those images that I showed you. So the results here are shown here on the top. So again, the same groups that we had before, controls, people with autism, the amygdala patient. Here's the eye tracking data that I showed you before. And here at the top, this is not, again, the eye tracking data. People are looking at the center of this image all the time. This just shows what information from faces they use in this bubbles task that I just described to you. So controls look at the eyes, and they use the eyes a lot. People with autism don't look at the eyes so much, and they use the eyes less. So you can see that in the difference here. This is the difference, control minus autism. Controls use the eyes a lot more. And the patient with amygdala lesions also uses the eyes less and looks at the eyes less. So the use of facial information actually is quite similar to where you look on the face, not surprisingly. So where have we been going with this? These are some, I just wanted to show you some recent data to give you a flavor of the studies that we're currently doing to extend this kind of work, which just uses pictures of faces, often very artificial stimuli, more to real-world kinds of stimuli. So we use stimuli that look like this. I don't know if the people in China are familiar with, or if it's popular to watch this episode from this series of episodes of The Office, which was very popular in the US. But what you have, people would be sitting there and we would do eye tracking, and there would be a 15-minute or so clip where you have complex social interactions. And there would be sound as well. I'm not playing you the sound. So this is an ideal stimulus. People have spent millions of dollars to create a maximally socially engaging stimulus, which is why people pay money to watch this. So it's all full of people and social interactions. And we can now ask, well, how do people with autism process this? And how do they look at this? This is a much richer stimulus and closer to the real world than these artificial faces. So if you wanted to do eye tracking, what you can imagine is you just have people watch the video and you do eye tracking. And so this little red cross would be where somebody looks while we do eye tracking during the video. You could do that. Here's a slightly different representation of the same thing. So this is where the subject would be looking throughout this 15-minute or so presentation of the video. But we don't have a single subject, so we have a group of subjects. So if you take a group of subjects and you ask how do they look, well, there will be a bunch of places where people look. So what this means is that most people looked here in the group. Maybe one person looked here, one person looked here, 4 or 5 people here, 3 people here, something like that. So you get some spatial dispersion, and it varies. Sometimes everybody looks in one place, and sometimes it gets much more spread out. So the actual data looks sort of like this. So as you have the movie going on, and you look at the eye tracking data from a group, sometimes everyone looks at the eyes, and then it spreads out and so forth. So you can quantify how normal, how similar to the group your eye tracking data are. So if we take one frame of this, so say we look up here in this frame, and we have a subject who fixated here. So this subject looked at a location where nobody else in the control group looked. Then we can plot that person's similarity to the group, and we can say, well, they're down here at 0, because nobody else looked up there. In the next frame, well, they looked over here. Most people didn't look there, but a few people looked there. So the similarity would be a little higher, up at 2. And then over here, they look over here, where everybody looks. So there, the similarity to the group would be very high. So you go through frame by frame of this video, and you compare where a subject looks relative to a normal reference group, and you get some time course of similarity over the whole duration of the movie. So that's the experiment that we did in a control group matched on these variables here, and a group, high-functioning group with autism. And it looks very similar here. So people with autism in red, controls in blue, similar eye tracking pattern. So the first point is to make is that there's lots of similarity. And this just plots each control subject's similarity measure over this whole video relative to that group. So here are the controls, here's autism. There is a little bit of a difference, group difference, but it's kind of swamped also by individual differences. So the question, though, that we're really interested in is, even though overall there's a lot of similarity in how people with autism look at a complex social stimulus like this video, and controls look, there's a lot of similarity, there are little points in time, which are these black bars, where there is a statistically significant difference between how people with autism and controls look. So sometimes people with autism don't look normally at this video. And what we would like to understand is what is responsible for this and what is happening at those particular times in the movie, to give us insight into what it is that's normally driving how we look at complex social stimuli, and what is it that's different in how the brains of people with autism process this. So just a couple of quick exploratory graphs. So these should be considered just exploratory and descriptive. But if you look at this similarity measure in viewing the video on the x-axis, and you correlate it with measures of social functioning. So in this case, this is just a social comprehension score. So after seeing the video, we ask people questions to ask how well did they understand it. And down here is poor understanding, up here is good understanding. So there's some correlation. If you have poor understanding of the video, you also have abnormal eye tracking. And there's a group separation as well, as you can see. So the blue is the controls, and the red is autism. And there's also a correlation with actual autism traits. So if you give people a questionnaire that measures autism symptoms, the autism quotient score, you can give this questionnaire to people with autism in red. You can also give this questionnaire to our healthy controls in blue. But of course, the blue controls have lower scores than autism, not surprisingly. But again, there's a correlation between this measure of individual differences in autism-like behavior and the abnormality of their eye tracking. So the eye tracking metric, the eye tracking similarity measure here that we're getting from this social video does seem to reflect social processing and does seem to correlate with autism symptoms. So what we're working on now is to ask, at what points in the movie, what is it that is actually driving people's social attention? And this is a very complex question because the movie is very complex. But what we're trying to build is sort of a full regression model of this complex social stimulus. The sort of semantic aspects of that model are very difficult. You have to have people rate the video. But the lower level ones can be defined objectively from just a computational model of saliency. So you can ask where on each frame are there particular color or contrast or brightness changes. And it turns out that there is a difference, a group difference there between controls and autism. The autism subjects look more, their gaze behavior is more driven by high contrast areas in the image than is the case for controls. So we're finding some differences there, and as I said, this is still very much work in progress, but ultimately what we would want to come up with is a full model that explains what are all the factors that make you look at a particular point, a particular location in that video. Some are low level, like if something is bright or something is moving fast. Some are high level, that you have a certain expectation of some person walking in, or there's something socially that's very interesting happening, and we want to see the extent to which these differentiate autism from controls. This is still work going on, but I just wanted to give you a flavor for the direction that we're heading. Let me switch gears here and zoom down to one of the most precise measures. So in this graph here of the resolution in space and time of different measures, so lesions and fMRI are up here, single unit recordings are by far have the best resolution in both space and time. They have a problem, which is that their field of view is tiny, but still they have very good resolution, which is why in many animal studies of social cognition, you want to put an electrode in and you want to measure how single neurons respond. We can do that in humans in very rare cases, and this is work that we've been doing for quite some time in collaboration with hospitals close to Caltech. And these are MRI images of 2 patients who have neurosurgical electrodes put into their brain. So this is a procedure that is done in this case for measuring epilepsy. So these are all people who have epilepsy, and they need to have part of their brain resected, taken the tissue removed that is causing the epilepsy. And in many cases, it's difficult to locate where that is. And so, for clinical reasons, they put electrodes into the brain, and the patients are sitting in their beds for a week or 2 weeks, and during that time, we can record from single neurons while we show the pictures. So this just shows the tracks of the electrodes here, and the tips of the electrodes sit in the amygdala. You can see it here as well. So this, just to orient you in this horizontal cut, so the eyes are up here, and the amygdala is here, and these black lines here are the electrodes that the surgeons have put in, from which we can record from the amygdala. Now this work has been done to some extent in humans by Itzhak Fried and Christoph Koch and colleagues, but also more extensively and more precisely in animals. And so this is work from Kati Gotthard at the University of Arizona. Just to give you one example, so these are single unit recordings from the amygdala in monkeys. And so if you record from the amygdala in monkey brains, you find that many of these neurons respond to social stimuli, to various kinds of body postures, to calls, and in particular to faces of other monkeys. And they can be very selective. So just 2 examples here. Here is one neuron recorded from the monkey amygdala, and this neuron responds only to a particular emotional expression, which is the expression of threat, regardless of the identity of the face that is shown. So these are 4 different monkey faces, but the threat expression drives these neurons to the amygdala. And over here is another neuron recorded from the monkey amygdala. This neuron cares about identity of the monkey face. This neuron fires strongly to this monkey, regardless of the expression, but less so to the other monkey faces. But so just an example that you find neurons in the primate amygdala that respond very well to faces. And so we did a similar study in human patients. So this just shows you these are 10, I guess, or so patients from whom you recorded. These are all the different colors, and these are the locations in the amygdala from which we recorded. So we can reconstruct from MRI images exactly where we record, and we know in each of these patients which region we record from. Actually, let me skip this slide. We showed them a lot of different kinds of stimuli. The details of that are not too important. But just to give you a flavor for how these neurons respond, so what we have here on the x-axis is time, and over here we show just a phase scrambled version of a face, and over here we show a face or parts of a face. That's what all these different colors mean. The details of that aren't important, but what this shows is that the face comes on, the face stimulus comes on here at 0, at this line, and then you see these neurons fire with a fairly long latency, about 300 to 500 milliseconds after the onset of the face. So each little dot is an action potential from a single neuron in the human amygdala, and this picture just shows that you get responses to faces or to parts of faces in the human amygdala as you do in the monkey amygdala. But to finish the story, what we did in a study that just came out is to record from people with epilepsy who did not have autism. That's all these blue locations, so 8 patients there. And we recorded from 2 patients who had epilepsy, but also had a diagnosis of autism. And those are those two red locations. And so this was a very unique opportunity to ask the question, if we record from single neurons in the amygdala in people with autism, can we find anything at the single cell level that tells us about differences in how those neurons might respond to faces? So the details of this picture aren't important, but what this just shows is waveforms, trough to peak ratios, and so forth, just basic electrophysiological properties of the neurons. And the story there is the neurons look normal in the autism subject. So they have normal waveforms, normal spontaneous firing rates, normal interspike interval histograms. There's nothing grossly abnormal about the electrophysiology of neurons in autism. But the next question that we asked is, well, how do they respond to faces? And for that, we return to this story that I showed you earlier, where we had this finding that the use of facial information, and also from eye tracking, how we look at faces, is abnormal in people with autism. Particularly people with autism don't seem to make use of the eye region of faces. So we used those same stimuli in these surgical patients. We showed them little bits of faces, these little bits of bubbles faces, while we record from single neurons in their amygdala. And we got a picture behaviorally that's similar to what I showed you before. So this is, you can imagine a face underneath here. Here's the eyes, here's the mouth. And in the controls, we got the same thing, that they make use of the eyes and the mouth, just like I showed you before. In the 2 patients with autism, we got, as I showed you before, that they use the mouth, but they have very little use of information from the eyes, just behaviorally from pushing buttons. But what we then did is we asked, well, how do amygdala neurons respond? So we can do the same analysis, not based on the behavior of the subjects, how well they can push the buttons, but based on the behavior of the neurons. That is, we show little bits of faces and ask which part of the face, the eyes, the nose, the mouth, is most powerful in driving neurons in the amygdala. And so in the controls, we find that neurons in the amygdala are also driven powerfully when the eyes are shown, and to some extent the mouth. But in the 2 patients that we recorded from in autism, over here on the left, you see a very different picture. So these neurons don't respond to the eye region of faces, but they respond very strongly to the mouth region. So we have a very crisp story that ties together a sort of high-level behavioral finding from eye tracking and this bubbles method, that people with autism don't seem to be able to make use of information from the eyes and faces. And when we ask where in their brain is that reflected, we see at the single neuron level in the amygdala that autism neurons respond differently. Let me switch gears again quickly, and this will be very quick, just to give you a flavor for where we're going at another sort of extreme of resolution here. So jumping up to fMRI and going back to this video that I had showed you earlier. So when people were shown this video that I started with and showed you the eye tracking data to, they were also lying inside an fMRI scanner. And so we could collect fMRI data while they're watching this video and ask how do their brains respond. And this is very difficult to analyze, very complicated, but so they would be lying in the scanner watching this. But I just want to show you some of the raw data to give you a flavor for the direction that we're going in. So if you look at early visual cortex, the fMRI signal in early visual cortex, people are lying in the scanner and they're looking at this, and you look just in the controls here at left or right early visual cortex, you find that there's a pretty consistent response. So the x-axis is time through this movie. They're seeing lots of patterns and things come by that stimulate early visual processing. Each of the thin lines is one subject's data, and the dark line is the mean from the control group. But you see that the signal time course is fairly similar. And same thing in early auditory cortex. They're all listening to this movie, and left and right auditory cortex look similar. And moreover, it looks that way if you compare the groups. So blue is the control group. This is how their visual cortex responds to the movie. So again, the x-axis is time. The y-axis is just a normalized version of the fMRI signal, the bold response in visual cortex. Each of the thin lines is one subject. The dark line is the mean. And this is controls in blue, and this is autism in red. And down here, to make it easy, I've just extracted these means. So you can see that they respond very similarly. And so, which is what you might expect. In fact, you can do this in monkeys, and you can find that early visual cortex in monkeys responds very similarly to what you see in humans. This isn't too surprising. So there's bright things happening, and darker scenes, and so on. So the input is the same to people with autism and to the controls, and early responses in the brain are also very similar early on. What we want to know is, well, what if we go to higher processing levels? At what point does the fMRI signal look different between autism and controls? And what does that tell us about the stage in processing at which people with autism might be abnormal? And so that's where we're currently sort of stuck. And so this will be the last slide on this experiment. But this is an example of a higher level region that's involved in empathizing, theory of mind, and so forth, medial prefrontal cortex. And if you take a look at the group means here, there is a difference between autism and the controls, but it's not statistically significant because each subject is very different. So you have big individual differences that swamp the group differences. So right now, the difficulty that we're facing really goes back to showing us that there are big individual differences in how people would interpret a complex social stimulus. And those are bigger, just in terms of differences between healthy individuals, than the difference between controls and autism subjects. So this is the current place we're at with that analysis. Let me wrap up here the last couple of minutes with another analysis which is much more common that people have done, and that is to look at what happens within a single brain in terms of relationships between bold signal in different regions, like functional connectivity that you saw some of this morning. So if you ask, just at rest, what is the correlation between the fMRI signal at one point here and the corresponding point in the other hemisphere, you will see that they're quite correlated. And in particular, homotopic regions in the left and the right hemisphere have a strong correlation. So you can show that, for instance, here in this correlation matrix. So if we take all the different brain regions, so these are segmented into these different colors. So you have early visual cortex, parts of the temporal lobe, and so forth. And each of those is a row here, and the same ones are also a column here in this symmetric matrix. And what this shows is the strength of the correlation in the bold signal between them. And so along the diagonal here, what you have is left visual cortex with right visual cortex, left primary motor cortex with right primary motor cortex. So every region on the left with its corresponding region on the right, and they're the most strongly coupled regions. So you have a strong coupling between the hemispheres. One study that we did recently was to look at the strength of that coupling also in people with autism. So there's a number of studies now claiming that this is reduced in people with autism, that they have abnormal, it is lower, long-range functional connectivity. And this rather complicated picture here just plots the difference between the strength of the left-right correlation in autism compared to controls. So the green ones here mean that the controls have a stronger correlation, and the group with autism has a weaker correlation. And this is just a histogram of those. So there's very few. It's very subtle. In particular, it's very subtle if you control for motion artifacts, which is what often hasn't been done. But there's some evidence in some brain regions that indeed there is reduced connectivity in the brains of people with autism. It's hard to tell with fMRI. As I said, it's very subtle, there's lots of motion artifacts. And so one thing that we did, this is the very last patient population here to give you a comparison to, was to ask, well, is there any comparison, like in the patient with amygdala lesions? We had a nice lesion comparison. Is there some comparison where we could tell this story with a particular patient group? And there is. And so if you look at bold signal between the left and the right hemispheres, that is mediated in part, of course it could be driven by common subcortical drivers, so the story is more complicated, but surely it's driven in part by the corpus callosum. And so that's shown here. If you dissect the dorsal part of a human brain away, you're looking down here at the corpus callosum. So this is the biggest white matter bundle in the human brain, and it observes long-distance connectivity between left and right hemispheres, about 200 million axons. Actually, surprisingly recent in its phylogenetic appearance. So it just appeared with placental mammals and marsupials like opossums and kangaroos that do not have a corpus callosum. Anyway, it's a huge fiber bundle in the human brain, and we studied this very rare population, people who are born without a corpus callosum, agenesis of the corpus callosum. So normally, here's a healthy subject, a sagittal MRI scan, here's the corpus callosum, those 200 million axons caught in cross-section, and here it's missing in the brain of a patient with agenesis of the corpus callosum. And you can reveal that if you do diffusion-weighted imaging, like the signal that people use for DTI-type measures. And this, you're looking down at a healthy brain here. Here's the front, here's the back. And these different colors encode the direction in which big white matter bundles run. So the corpus callosum, so red is left to right. And the corpus callosum is this big red region here where there's all these fiber bundles are oriented left to right. Completely missing on the right in the brain of a patient with agenesis of the corpus callosum. So they have a very interesting phenotype. I'm not going to spend any time on this slide here, but this summarizes for you some of the deficits that they have. It's complicated, but the bottom line is that about a third of them meet criteria for autism. And we've actually just quantified this in a paper that hopefully will be out soon. It's difficult to estimate this because the patients are very rare. So the incidence of agenesis of the corpus callosum is about 1 in 4,000, but we've studied about 50 of them in the lab and compared them to autism. And if you do all the standard diagnostic, use all the standard diagnostic instruments, the Autism Diagnostic Observation Schedule, the Autism Diagnostic Interview, the ADOS and the ADI, as well as clinical interview, about a third meet criteria for autism. So this is way higher than in the typically developed population. So there clearly seems to be a link between the corpus callosum and autism. So this comparison between these clinical groups together with the fMRI data that I showed you lends some support to that hypothesis. And let me just end up in the last two slides here by showing you some future directions that I think are interesting to go into. One is that there is a mouse model of autism studied by Jackie Crawley, amongst others. So this is a mouse that, like in our human patients, is born without a corpus callosum and has social deficits that in many respects seem to be a model for autism. And this just shows DTI on some of the white matter tracts. So like in the human patients, these mice don't have a corpus callosum. Instead, what they have is these weird fiber bundles that a healthy brain does not have, called Probst bundles, that connect each hemisphere within a hemisphere, but they don't cross over. So there's an animal model that, of autism that fortuitously also had agenesis of the corpus callosum. So sort of by coincidence, there is that convergence between autism and callosal agenesis also in an animal model. The final one, which is something we've been trying to do, but it's hard, but it turns out that, so I told you autism, you cannot diagnose at birth. You don't know if a baby will go on to develop autism. You might know about some of the risk factors, but you don't know until about age 2 or 3 when you can actually diagnose autism. And most people now working on autism are trying to understand what happens before age 2 and 3, what happens very early on in development, and maybe if we understand that, we can try and influence early development and change the developmental trajectory. Well, these patients with agenesis of the corpus callosum, you can tell at birth. In fact, you can tell before birth, because nowadays this is an ultrasound image of a pregnant mother, and the fetus in the womb here, not to you or me, but to a radiologist, a gynecologist looking at this image, you can diagnose absence of the corpus callosum in utero before birth. And so there would be a population of babies that will be born that we would already know have absence of the corpus callosum, and we could follow their brain development and their behavioral development from birth, or to some extent even before birth. And we know that 1/3 of them will go on and develop autism. So that is one model that we're trying to work on. And let me end there by acknowledging the various funding agencies that have made this work possible, and then people in my lab and several collaborators. Michael Spezia, who's now a professor at Scripps College, did a lot of the early eye tracking work in autism. Owana and Shuo have done lots of work with the intracranial recordings. Lynn Paul is a person who's been in my lab for a long time and used to be the president of the only international organization for people with agenesis of the corpus callosum, NODCC, National Organization for Disorders of the Corpus Callosum. This is Lynn Paul. And then Dan Kennedy, who used to be a postdoc with me, now faculty at Indiana University, has done a lot of this work using the movies and autism. And then finally, there are 2 hospitals, Huntington Memorial Hospital and Cedars-Sinai Medical Center in Los Angeles, where we, with these neurosurgeons and this faculty person there, who also used to be a postdoc, Uli Ruteshauser, that's where we do the intracranial recordings in the surgical patients. And finally, Joe Piven at UNC, with whom we collaborated on all the autism studies. And if you want more information, there's a couple of different websites here that give you information about several of the research projects and a lot more than the ones that I talked about. And also a graduate program at Caltech on social neuroscience that's listed up here. And let me end with that. Thank you. So the question was, what's the frequency of agenesis of the corpus callosum in humans? So it's quite rare. It's about 1 in 4,000 is the estimate. But that's, sort of all people with agenesis of the corpus callosum. Once you put in the exclusionary criteria that we have, which is that they need to be high functioning, they need to have no other developmental abnormality, it's going to be a lot rarer than that. I have no idea what it would be, maybe 1 in 20,000 or something like that. I should point out also that one thing that I didn't mention, so these are, they don't have a corpus callosum, these patients, but they're quite different in their presentation from the extremely rare surgical split brain, so-called split brain patients, right? Surgical resection of the corpus callosum for treating epilepsy. So those were people who had a normal corpus callosum, and then in adulthood that was sectioned surgically. These people here, they never developed a corpus callosum in the first place.
So with it being that, seems like it would be difficult obstetrician.
So
do you have some way of predicting who might be at risk?
Yeah, so Larry's pointing out obvious difficulties with this last future research direction, which I pointed out. That's certainly one of, that's not the least of the problems, but right, it's quite difficult. So given, it would always be diagnosed, but you would have to have the sort of gynecologist, really a big network of gynecologists plugged in order to detect it, given the relatively low base rate that you would have here. So we do have a few, not fetuses or mothers who are pregnant with people with absence of the corpus callosum, but we do have a few very young children. I'm trying to think what the earliest age is, maybe age 2 or something. So we have some very early infants, but indeed detecting it in utero and being able to actually do measures that early is challenging. Low base rates, you have to have a big network. If you do the calculations, you would definitely, if you had a nationwide network for this, you would certainly be able to get some, but it would take some logistical problems to set it up.
I follow on from that. I mean, the callosal agenesis is also being associated with schizophrenia by a number of studies, albeit with the same caveats. It's a very small population. But on top of that, there are a number of studies showing sort of disconnection between the hemispheres as being very strongly associated with schizophrenia, more so really than autism.
Right. So the point there was that agenesis of the corpus callosum can be associated with, in adulthood, a number of psychiatric illnesses. And I think it brings us back to this more dimensional account, that we would want to really assess these patients in detail in a number of dimensions, and then ask the question, what pushes them in one direction or the other? Those that go on to develop autism, those that look more like schizophrenia, and so forth. We've tried to do that. So of the 50 that we have, we have assessed them in great detail, but mostly with respect to autism and sort of social cognition dimensions. We don't have an answer to that. We've tried hard. So we, for instance, had a look at the hypothesis of whether complete agenesis of the corpus callosum versus partial agenesis, which you also sometimes find, whether that's a factor, and it doesn't seem to be. So right now we haven't found anything that differentiates those that go on to develop autism from the 2/3 that don't.
So for the study, you just mentioned that fMRI activity when people watch a movie. I'm curious about that. It only shows 3 areas. And you know, that's the whole brain. I mean, you defined some RIs, or did you look at the whole brain? Particularly, you didn't mention activity from amygdala.
Right, so the question was, in the preliminary data that I showed you of fMRI responses to the movie, I only showed 3 time courses from 3 example regions. But of course, we have a whole brain field of view, and indeed we're looking at the whole brain. The reason I didn't show it to you is because I don't have any particularly clear story yet to present here. But you're right. Of course, that's what we want to do. We want to look at the whole brain and ask, how does the whole brain respond? There are many different ways of looking at that. One direction that we're taking, that some other people have taken also, is to take the brains of all the different subjects in the group and to correlate the time courses of the Bolt signal between those different brains at each region. So you can ask, how similar are the brains responding? And it's very similar to the eye tracking data analysis that I showed you. You can ask, how similar all over the brain are the brains of individuals in the group responding in a control group and then an autism group. So we're looking at that, and we're finding some differences, but it does come back to the same problem that I showed in this little example here, which is there are many brain regions where you find things are very similar in controls and autism, and then there's some regions where there are differences, but it's very difficult to attribute those differences to autism, because you also find differences between people. It's not too surprising. So if you take a look at this movie, The Office, and you watch it and I watch it, you might find some parts really funny, and I find them not funny at all. So surely you would find differences in the brain. So accounting for those individual differences and incorporating those into some kind of quantitative model is sort of the place where we're at right now in our research. But you're absolutely right. We have whole brain data, and indeed we're analyzing the whole brain, not just parts of it.
Thanks.
Thank you very much.